So the first section is the human microbiome in health and disease. So planet-wide, bacteria are exceptionally abundant. In rich ocean waters, like the ones off the coast of New Jersey, there's something like 10 million bacteria per mil, a million bacteria per mil in nutrient-rich waters, which multiplies out to something like 5 times 10 to the 30 bacterial cells on Earth. So you could say the story of life on Earth is a story of microbes and a little bit of other stuff. And that certainly connects as well with human welfare and the human microbiome. So microbes tend to live in mixed communities. These vast numbers of bacteria are often different ones living together. And often it's the interaction of all the components of these communities that result in the sort of phenotypic output or the consequences of bacterial colonization. For example, the material that's excreted from one type of bacteria may be food for the next one. So it's the action of the whole community that influences the inputs and outputs. So this picture just emphasizes that. My student, Arwa Abbas, really likes manga illustration. So this is manga microbes, just illustrating that bugs tend to live in communities. So as you very well know, we can now read out the composition of these communities and some of the aspects of function using DNA sequencing. And so the growth of the ability to sequence DNA has been nothing short of astounding. I'm old enough to remember using radioactively end labeling DNA molecules and running these long sequencing gels. And if you could read out a few dozen bases, you thought you'd done a pretty good job of it. Today, with the new Illumina HiSeq instruments, you can genuinely collect 1 billion bases of sequence information in a single instrument run, which can approach 500 billion bases of sequence total output. 1 billion reads and 500 billion bases of sequence output. So just unbelievable amounts of sequence. And so you can use that to characterize mixed microbial communities and read out stuff about them. So to say a little more about that, this area has come to be called metagenomics. So the idea is that if you have one of these complex mixtures of bacteria and other microbes, you can collect a bunch of samples, maybe from healthy people, collect a bunch of other samples, maybe from sick people, sequence deeply into each sample, and often there's so many different kinds of bacteria that you're not going to sequence every genome in the sample, but you can get a lot of sequence information that you can align reads to databases, read out the kinds of bacteria that are there, read out the kinds of genes that are there, and then use that sequence sample to make inferences about the disease state or colonization in a certain setting or what have you. So there are various ways to go about this. These sorts of studies have come to be called metagenomics. One way is to use tag sequencing. So turns out for bacteria that the 16S ribosomal rRNA gene has some highly conserved regions in it. So that's handy because you can land PCR primers on those highly conserved regions, PCR amplify, then sequence the bit in between, and then by aligning to databases, you can read out the kind of bacteria that's there. So if you have a mixed community with a whole bunch of different kinds of bacteria, you can PCR amplify, sequence these 16S tags, and count the number of reads you get for each kind. That gives you a rough idea of abundance and allows you to tell who's there within that community. But that's only allowing you to look at the bacteria and only giving you sort of a who's there kind of snapshot of the bugs that are there. A richer picture can come from shotgun metagenomic sequencing. This shows a bacterial genome, one of the smallest, from Mycoplasma genitalium. Imagine shredding up the DNA of this bacteria and lots of other bacteria in a complex mixture, and then sequencing lots and lots of those little pieces. So you would read out genes important for adenine biosynthesis, for ribosomal proteins, for antibiotic resistance, whatever, all in some giant collection. And also you would be able to assign the closest known relative to that gene. Is it Bacteroidetes fragilis or Escherichia coli or what have you? So you can get quite rich information on the composition of that community, although it's much more of an analytical challenge and more expensive to generate this kind of data. So one wants to kind of pick one's battles in choosing between the relatively simpler tag sequencing approach and the shotgun metagenomic approach. So the vertebrate microbiome, as you probably well know, is a complex mixture of prokaryotes, archaea, eukaryotes, and their viruses. The human GI tract is home to some 100 trillion bacterial cells and their associated viruses preying on those bacterial cells and archaeal cells. The density of bacteria in the human colon is something like 10 to the 11th to 10 to the 12th per mil, just enormous numbers of bacteria present in gut. The gene number in the microbiota, if you pile up all of these bacteria, is at least tenfold greater than the number of different kinds of genes in the human genome itself. There are many species of each of these forms present associated with humans. Most have not been cultured outside of the human body, so they're not well understood at all. And they're distinctive microbiota at different body sites. That is, gut is different from skin, different from airway, and so on. And so here's a cluster map. I'll say more in a minute how we generate these kinds of pictures. But you can see samples from oropharyngeal, that's the purple, from red, from nasopharyngeal, red, and fecal samples, blue, all in this sort of cluster diagram where proximity means more similar, distance means more different. You can see that these samples cluster by body type, despite the fact that they're from lots of different human individuals. And this is just a little bit of data illustrating that they're distinctive microbiota at different body sites. Now, of course, the reason for being interested in this, in large measure, is the associations with human disease. Of course, it's well understood that there are many infectious agents that directly infect humans and cause disease, and many of those are well known, though probably not all of them. These metagenomic methods have been used widely to hunt for new bugs in different disease settings. But the microbiome, or the full population of bugs associated with the human body, has been implicated as a modulator in many disease states, maybe all of the major disease states at this point. So oops, well, that was a surprise. So atherosclerosis, for example, has been linked to microbial colonization in several respects, oral gut and plaque in the microbiota, microbial metabolism of choline and carnitine to yield TMA, a very interesting proposal from recent data from Stan Hazen. Colon cancer, several proposals for involvement of specific lineages and the general contributions of dysbiotic communities. IBD, dysbiosis, is widely believed to be an important contributor, and I'll say more about that as we go along. And it may have an infectious trigger. Asthma, it may be the lack of bugs according to the hygiene hypothesis. Diabetes, several specific proposals. Autism, some very interesting suggestions that bacterial co-metabolites exposed to the fetus during gestation may influence the development of autism. The microbiome, as I'll show you in a minute, is a contributor to drug metabolism. Has been shown in several different states, several different agents, and many, many further proposals as well. Many of these are modulators where the bugs that are associated with your body are maybe having one of many contributors to developing the disease state, and others that suggest that the action is more direct and quantitatively more significant. So a whole list of suggestions basically associating the microbiota, at least as a proposal, with most or all of the major diseases that are affecting Westerners.